Okay, so hello everyone. Welcome to our brain-body interaction seminar series. We are happy to welcome Professor Julia Cordero today and all of you for your presence. Just general information before we start. Our series happen on Tuesdays around 4 p.m. UK time, where we try to bring together scientists interested in the brain-body interactions field. You can check our series on our website, which is brainbodyinteractions.org. And there you can also subscribe to our mailing list to get our weekly emails about our talks. And there you can also find our previous seminars. So feel free to go and get to know our previous speakers. So it is my pleasure to introduce Professor Julia Cordero today. Professor Julia Cordero obtained a PhD degree in molecular and cellular biology program at Washington University School of Medicine under the advisorship of Dr. Ross Al-Kagan. She stayed in the lab for a one-year postdoc and moved on to a postdoctoral fellow position at the Beatson Institute for Cancer Research in Glasgow under the advisorship of Professor Owen J. Sanson. Dr. Julia Cordero is since 2022 a professor of systemic signaling biology at the Wolfson Cancer Research Center at the School of Cancer Sciences at the University of Glasgow. And since 2016 until now, she's an honorary group leader at the Beatson Institute for Cancer Research at the Institute of Cancer Sciences, Glasgow. And her lab is devoted to understanding how intestinal stem cells adapt and respond to changes in the micro and macro environment, how the intestine senses and controls whole body homeostasis, and how intestinal dysfunction can lead to broader organismal instability. The lab uses the fruit fly Drosophila melanogaster because it's an amazing model organism, but because it's a primary research model system due to its unparalleled genetic power and amenability for multi-organ in vivo studies combined with experiments with mammalian systems. And today she'll be telling us about gut-body interactions in health and disease. So if you have questions, feel free to drop them in the chat box and we'll get to them towards the end of the talk. So with this, I give the stage to Professor Cordero. Thank you.
Okay, can you hear me well, Rita and everyone? Okay, thank you so much. Rita, thanks a lot for inviting me to be part of this really wonderful series. Thanks to you and the organizers. And I hope, yes, it's more of a gut-centric than a gut-brain thing, but there is a little bit of brain. So hopefully people will not be too disappointed. My lab has been interested in studying adult intestinal biology for a while, and that's what I started with my postdoc, really. And so we are really interested in understanding how, gut disease, how gut health is regulated, how gut disease can, impact the normal functions of the gut, but also the whole organism, as Rita mentioned in her introduction. And we really see the gut as really a master regulator of organismal health, and it's a well-known tissue because it really regulates essential life functions like the epithelial barrier and protecting our body from external agents that can, damage us. It's also has essential immunity functions. It's a regulator of metabolism. The gut is the biggest endocrine organ in our bodies, and it also regulates higher order functions through the gut-brain axis. So really, this emphasizes the importance of understanding how gut the healthy status of this tissue is maintained, but also it makes us realize how important keeping the health of the system is, because you can imagine that an organ that has so many important functions, when it gets sick, when something goes wrong, then it will have implications beyond the health of its own system, and it has implications to the whole body. And the system that we mainly use, the model system that we mainly use to study gut biology is the fly, the adult fly. The adult fly gut has this very much the same physiological functions that the mammalian's small intestine, and we focus on the midgut of the fly. Not only that, it has the similar cell types, and also the cellular and the molecular mechanisms that regulate biology and physiology and metabolism in the adult fly midgut are very much conserved to those in the mammalian system. So the fly gut does really represent an amazing model system where we can study biology of the intestine and we study the intestine as part of a multi-organ system in vivo, and also within its natural microenvironment. So it's a very amenable system, and these are the type of questions that the reasons why we are really love using this system. However, it's really exciting as well for us, and when we can, we try to translate some of our discoveries made in flies into mammalian settings when we can. By mammalian settings, I mean mouse models or intestinal organoids, so on, depending on the question that we want to ask. So the adult fly midgut is a monolayer epithelium. It's a simpler sort of architecturally compared to the mammalian system, but it does contain all the different cell types that the mammalian gut has. It has, very importantly, intestinal stem cells, and that are essential to give this organ the regenerative capacity, and also the capacity to be transformed, to undergo malignant transformation. Really, these intestinal stem cells would give rise to an undifferentiated progeny, or a more naive progeny, which is the enteroblast, which is sort of an intermediate progeny in the lineage. But this enteroblast, or another, there are different types of enteroblast, but just to simplify, enteroblast would then give rise to the differentiated cell types in the gut, which are the enterocytes and the enteroendocrine cells. Enterocytes being the main component of the intestinal epithelium, the most predominant component of the intestinal epithelium, which has absorptive and metabolic functions that are essential for our body, and also the enteroendocrine cells, which have cells that have sensory and secretory capacities, and they are the cells that really make the intestine the biggest endocrine organ in our body. So we do have very nice genetic tools in flies, as you very well know, and in the case of the gut, we have different genetic tools to visualize and genetically manipulate all the different cell lineages in the gut, but also gut-associated cell types. This is an example of a very popular driver of intestinal stem cells and enteroblast that you can see here. You can label these cells, and you can see them in green. We can also have similar tools for enterocyte visualization and genetic manipulation, and similarly we have enteroendocrine cell drivers that we use as well. Now, there are different paradigms that we use in the fly to study, the questions of interest, and one of the important characteristics that the fly gut has, which is concerned with the mammalian system, is that it's the capacity of this tissue to highly regenerate very, very actively in response to external stimuli or external challenges. And for example, if we feed flies with chemicals or bacterial pathogens, we can cause damage to the intestinal epithelium, and that basically mainly causes damage to the enterocytes in the gut. This damage will generate signals that are sensed by the stem cells and drives the proliferation of the stem cell, and this proliferative response to the stem cells to damage the epithelium is essential to regenerate the damaged tissue and also to protect the organismal integrity in response to these challenges. In real life, a regenerating gut looks like this. After challenging the system with chemicals or pathogens, the stem cells proliferate. We see an expansion of the stem cell compartment, and we can quantify this proliferation by staining the gut, for example, with an antibody for phosphohistone to label proliferating intestinal stem cells, and then we can have a very clear quantitative measure of gut regeneration. We can also drive colorectal cancer-like phenotypes in the fly gut, so we can transform stem cells in the fly gut to using oncogenes or tumor suppressors that would are normally mutated in the human disease. And by doing this, we can recapitulate various aspects of colorectal cancer or inflammatory diseases in the gut. These are different genes that are associated to human diseases that when we either downregulate them or overexpress them, depending on whether they are tumor suppressors or oncogenes in the stem cells, we can drive a very strong hyperplastic response in the gut, which is also manifested by an expansion of the stem progenitors domain, increasing the proliferation of stem cells, but many other manifestations. And this is just an example of how, what happens to a mammalian intestine when you do similar genetic manipulations. You also see this expansion of the stem cell domain that you can see here in green. And we have worked with these various models of either hyperplasia or gut regeneration over the years and in the lab. And these are things that we continue to work with and use as paradigms to study stem cell biology in those contexts. So there are various research questions that we pursue in the lab, which are sort of interrelated in many different ways. So one of the things that we are interested in understanding is, how these challenges to the gut caused by either damage, inflammation, or the presence of tumors, what are the local responses to this type of challenges? And by local response, we mean the interactions, how these type of challenges modify, the interactions, signaling, and crosstalk between the gut and its microenvironment, meaning, tissues that are closely associated to the gut, how these interactions also in turn modify the adaptations or drive adaptations and responses of the stem cells compartment to these challenges. And also, we look more systemically to how, changes in the gut caused by different stimuli, like indicated here, how these changes and this disruption to a normal homeostatic status of the gut can lead to broader manifestations in our bodies or systemic responses in our body. So I will do sort of two parts to the talk today based on the research that we are currently on in the lab. And just to say, all the work that I'm going to show is unpublished. So please keep this, I know this is being recorded and that's fine, but, just to make a statement that I'm showing all unpublished data today. And I would like to, I'm going to start with the gut niche interactions as a first part of the talk. And in particular, at the studies we've been doing for a few years on the interactions between the gut and its vasculature, and how the interactions between the gut and its associated vascular tissue impacts homeostasis of the gut, or really the adaptations of the gut to changes in the environment. And the gut, the mammalian intestine is highly vascularized. And similarly, the adult fly midgut is very prominently associated with gut-associated tracheal tissue, which is akin to the mammalian vasculatures. And the gut tracheal tissue here, you can see it in red. So this tracheal system in the flies is a system that is in charge of delivering oxygen to the gut, in this case, or to any target tissue, but also exchanging signals and metabolites and so on with the target tissue. So it does fulfill many of the functions that the vasculature does. Now, in spite of a very well-known fact that the intestinal vasculature is a very important and prominent component of the intestine, very little was known about how these tissues interact with each other and how whether this physical association does have a role in the context of homeostasis or tissue regeneration. And these were things that we're very interested to understand. And the work that I will talk about regarding this project has been entirely driven by Jessica Perochon, a postdoc in the lab, together with Jade Phillips, a PhD student. And so this project started with an observation that Jessica made on the fact that when we induce regeneration of the gut, we induce damage in the gut by feeding animals with these pathogens or with chemical agents and causing this local damage to the gut epithelium. That turned out to lead to a very prominent response, very strong response of the not only the stem cells, which we know proliferate in response to damage, but also to the associated tracheal tissue, which is not directly damaged, but is regardless responding to the gut damage by remodeling very significantly. And this is like a terminal tracheal cell similar to tip cells in the mammalian vasculature that extends many projections in response to during the regeneration of the gut. And this phenomenology of tracheal response to gut damage was also conserved in the mammalian system. So this is a small intestine of the mouse that is in homeostatic conditions or following damage by whole body irradiation. And what we can see here are regenerated crypts of the mouse small intestine. And in brown, you can see CD31 labeled endothelial cells of the vasculature. So you can probably appreciate that there is a much more prominent signal of endothelial cells in regenerating crypts versus in the control crypts. So this we were excited to see that this phenomenology that we observe in the gut-associated tracheal system was recapitulated in the vasculature in the mouse. So we did a lot of work to quantify this type of remodeling of the tracheal system in the gut, and we could observe that what happens in this case is when there is a regeneration of the gut, these tracheal cells, individual tracheal cells, do increase the number of and the length of their cellular projections into the tracheal system. There is no more number of cells, but there is an active remodeling of these cells, which is quantified here. And this actually is sort of schematizing this here. So then we use genetic tools to ask the question as to whether this tracheal remodeling was functional to the regenerative proliferation of the gut, the regenerative proliferation of the stem cells in the gut. And so to answer this question, we use a tracheal-specific driver, and we drove a pro-apoptotic gene BAX, which you can see the outcome here, right? Well, driving this pro-apoptotic gene leads to a very strong reduction in tracheal cells that are associated in the gut. And then we induce the gut to regenerate, and we ask, so what happens to the proliferative response of the stem cells in this case? And surely enough, we saw a very significant impairment in the proliferative response or the capacity of the gut to regenerate in response to damage when the tracheal system is not fully active or not fully able to remodel and provide, whatever signals it's providing. So there is an important role for this associated tissue to the gut regeneration. And so I'm going to summarize here many years of work that led to the initial working model in this project. Basically, what we found was that the production of reactive oxygen species from the gut, which are made as a response, an initial response to the pathogenic damage, is a signal that is important to trigger changes in the tracheal cells. These changes involve the activation of multiple signaling pathways and genes, basically a very interesting molecular program including the activation of HIF-1 alpha, FGF receptor signaling, but also the activation of unexpected genes like neuronal remodeling genes, the downregulation of embryonic tracheal specification factors. But this program was very important to drive the remodeling of this tracheal tissue. And in turn, this tracheal tissue remodeling also is necessary and part of a process that leads to oxygen and robust oxygen delivery to the gut, but also the production of angiocrine factors. For example, FGF receptor, which actively binds to the FGF ligand, which binds to FGF receptor in the stem cells, and then the stem cells proliferate in response to that signal. So there is a reciprocal crosstalk between the gut and the tracheal system, in this case, driven by chemicals and secreted factors that are conductive to the robust not only remodeling of the tracheal system, but also the regeneration and of the gut in response to damage. However, during the course of this work, we made some observations that indicated to us that there was more to this model, as it usually happens, more than what we were had been able to figure out. And one observation that was very interesting to us was to see that when we have damage in the gut, but we prevent the enterocytes or the epithelial cells from dying, which is part of the response to this damage. But it's a response that lies downstream of the production of reactive oxygen species. So this prevention of cell death of enterocytes was sufficient to impair the remodeling of the tracheal cells and to also block regenerative proliferation of the stem cells. And the data to support this is shown here. So this is in red, you can see the tracheal system, in blue is the gut. So this is a control gut with homeostasis and following damage, lots, big increase in the tracheal associated tissue. However, if we block, if we induce, we prevent apoptosis of the stems of the enterocytes by overexpressing an anti-apoptotic gene, we can see that this remodeling in response to damage is suppressed. And this is quantified here. So in itself, the death of the enterocytes is an important trigger to this remodeling of the trachea. However, this is a context where we still have high oxygen, reactive oxygen species made. But furthermore, we have observed, and we reported this in our initial publication, that this remodeling of the tracheal cells in response to gut damage was not only happening in the context of pathogenic infection, but also, for example, in the context of the damage by DSS. And this is quantified here. Now, damage by DSS is not sensitive to, and that does not lead to the production of reactive oxygen species. And consistent with that, if we treat DSS damaged guts with antioxidant NAC, the tracheal remodeling is not affected by this. So altogether, this evidence suggests that there are gut signals other than or in addition to reactive oxygen species, which also are influencing tracheal remodeling. So, you know, there are more than just this chemical signal influencing the crosstalk between the gut and the trachea. And, you know, to start investigating this phenomenology and following on this observation further, we went back to some of our initial electron microscopies of this system, and that showed that terminal tracheal cells, the extensions, these projections, these long cellular projections of terminal tracheal cells are in very close physical contact, direct apparently, but we didn't prove it at the time, physical contact with the intestinal epithelium. And this is an terminal tracheal cell projection. This is the basal lamina, the visceral muscle, and this is the gut epithelium. This is an enterocyte. So you can see that there is even like these terminal tracheal projections have invaded through the basal lamina and into the gut epithelium, suggesting the possibility that indeed these two cell types are in direct physical contact. And then to to prove whether this is indeed the case, what we used was the GRASP system, which originally developed and used most popularly in neurobiology to identify synaptic partners. And so we drive a part of a GFP with a driver from one cell type, and the other half of the GFP with a driver of a different cell type. And only if these two cell types are in close contact, then the GFP gets reconstituted and we see a GFP signal. So we use this system to drive these two halves of the GFP in the gut epithelium or the tracheal cells. And in red, you would see a tracheal cell because it's a constitutive marker like a RFP driven by this tracheal driver, but there is no much GFP signaling in homeostatic conditions of the gut. However, when we induce damage to the gut epithelium, we could see then that there is an increase in the number of GFP positive cells, and these are enterocyte cells that we observe in the gut, suggesting that there is an inducible, at least temporary physical contact between tracheal cells and the gut epithelium during the context of gut regeneration, suggesting a direct physical interaction between these cells. And this is quantified here. So furthermore, we observed that the changes in tracheal remodeling that occur during the process of gut regeneration and also the recovery of gut homeostasis following damage is very dynamic. So if we look at tracheal remodeling over time, and I'm here showing you a trace of a single tracheal cell in white, you can see that in homeostasis and in the recovery phase of the gut, this is after we have had damage removed, then the cells look pretty much the same, whereas you have active remodeling during the damage. So there is a reversible process, this remodeling of tracheal cells, which accompanies very significant changes in gut shape and gut size that are characteristics of these different stages of the gut regeneration process. So during the damaged phase of the gut regeneration process, the posterior midgut of the flies becomes shorter in the anterior posterior axis and thicker or longer in the dorsoventral axis. And this changes in shape is reversed during the recovery period. And if we look at the epithelium of the gut during these different phases of the regeneration process, and here we have basically used markers of junctional markers for the enterocytes and intestinal stem cells and look at this over time, and we can trace individual epithelial cells, enterocytes here. You can probably observe that during this process, these different stages, during the damage stage, the enterocytes become more oval, and this is restored. This shape change is reverted to a more circular shape during the recovery phase. And so Jade in the lab did a lot of imaging and segmentation of the gut epithelium during these various stages, and she measured what we call eccentricity, which is essentially the level of the loss of the round shape for enterocytes. And so the more light yellow that you see, the more eccentricity the system has. And so we measure eccentricity during the different stages, and we saw an increase of eccentricity during the damage phase of the recovery of the regeneration process, which is reversed during the recovery. And interestingly, if we play with when the death of the enterocytes and during this process, we observed that the death of the enterocyte was a major driver. It was necessary and sufficient to drive these eccentricity changes, suggesting that this phenomenology we saw the impact of enterocyte cell death in the remodeling of the trachea has something to do with actual changes that are happening and the architecture of the gut epithelium during the process of regeneration. So then we ask whether then can we recapitulate these changes in tracheal remodeling if we simply play with the eccentricity of the enterocytes, or if we simply play with the shape of the gut rather than inducing damage. And to answer this question, we fed the flies with methyl cellulose, which is a polymer that the flies can eat but not digest, so it will inflate their bellies and their guts. And feeding this polymer leads to changes in gut shape that are similar to those that we observed during the homeostatic versus damage phase of the gut regeneration process. And then we measure eccentricity in this context, and then we see that, in fact, the guts that have been, from flies that have been fed methyl cellulose show more eccentricity in their enterocytes, so we can recapitulate these eccentricity changes that we saw in regeneration by using this approach. And in this case, we can also, this is sufficient to drive remodeling of the tracheal tissue. This is without reactive oxygen species production, without any sort of damage that we can detect. So suggesting that this stretching, the mechanical stretch that is caused in the gut epithelium during the process of regeneration is an important trigger to the remodeling of the gut-associated trachea. And this is summarized here. Now, what are the signals? How is this mechanical signal transduced? What are the mechanisms that drive, this mechanically induced crosstalk between the gut and the trachea? And we have started to address this question. So the way that we thought to go about this is to start with single cell RNA sequencing of terminal tracheal cells. Actually, tracheal cells, this is a driver that would label the entire all tracheal cells in the gut and in the flies, but we would look at control or following damage of the gut, and then we dissect the gut and we select tracheal cells specifically by sorting them by RFP positivity, and then we do single cell sequencing to identify, what could be molecular changes that are happening beyond the ones that we already knew in this tracheal system. And first thing that we saw was that these adult tracheal cells from the gut are highly diverse. There is not, there are at least, we detected at least 7 different subgroups of tracheal cells in the gut. And we found that there is a small subtype of the tracheal cells within subgroup 5 that express the mechanosensory channel piezo. And you can see here, if we look at the expression of piezo, is highly overrepresented in the context of infection, which is in the regenerating gut. Yes, so there is an induction of this expression in this of this channel in a subpopulation of tracheal cells, which is happening in the context of regeneration. And then we use a reporter of piezo gene expression in the trachea, like endogenous piezo gene reporter expression, and we combine this with a total tracheal reporter, and we could observe that here in GFP you have piezo, and here is all the trachea, and indeed there are some tracheal cells that do express piezo, like this one here, but there are other tracheal cells that don't seem to have, at least they don't have as detectable levels of piezo. So there seems to be a diversity of tracheal cells, and at least some cells express this mechanosensory channel. So then we went back to our different phases of the gut regeneration, and we used the piezo gene reporter to look at expression over time. And what we could see is that this plasticity that we see in the trachea, the remodeling, this reversibility of the trachea and the changes in the gut, the shape over these various stages, also is accompanied by a very dynamic expression of piezo gene in the tracheal system, which is highest during the damage phase and then returns to homeostasis in the recovery phase. So piezo is a mechanosensory channel and is activated. It's a calcium it's a channel that leads to calcium influxes in the cell, and it's activated by mechanical forces, membrane deformation. And so we wanted to see whether, the signaling by this channel was activated in the tracheal cells in response to damage. And this is what I'm going to show you here, if the movie is played. So these are movies that are constantly running, but basically this is a calcium sensor, GCAM7, expressed in the tracheal cells of control or regenerating guts. And this is ex vivo imaging of these guts. And as you can probably appreciate, there is very significant calcium waves in these tracheal cells in the regenerating gut, but not much at all in the control gut. Now, if we use an RNAi to knock down piezo specifically from tracheal cells, and then we look at these calcium waves, then we can completely see there is almost a complete disappearance of this calcium waves, suggesting that indeed this channel is being activated in the tracheal cells in response to during the regeneration of the gut. And these calcium waves are, specific, at least as far as we can tell from this channel activity. It's a good readout of this channel activity. And next, we did functional experiments to assess whether, is the expression of piezo in the tracheal cells important for regeneration of the gut? And so we knocked down piezo specifically in terminal tracheal cells during the adult system, and we use 2 different independent RNAi lines, and we could see that, in fact, knocking down piezo in tracheal cells prevents tracheal remodeling in response to damage of the gut, but it also impacts on the regenerative response, the regenerative capacity of the gut as per our measures of intestinal stem cell proliferation in the different contexts. So altogether, what we can say is that in addition to chemical signals, there are mechanical signals that work in parallel to activate tracheal remodeling in the context of intestinal damage. And these are important triggers to drive the remodeling of the trachea, but also necessary triggers to then also drive robust regeneration in the gut and stem cell proliferation as part of this regeneration. So now we are working further to identify what lies downstream of piezo, whether there are common signals to the chemical ones or whether there are distinct signals. And, some of the questions, those are some of the questions we are trying to address. But also we are moving some of our regeneration experiments from regeneration to tumor models, because we can also very nicely not only image the process of gut and tracheal interactions in the context of regeneration, but also in the context of tumors. This is an example of cells that are expressing the oncogenes, stem cells that are expressing the oncogene SARC, and we can look at them over time. You can possibly in green maybe see that over time you see an expansion of the stem cell domain. We also can in parallel look at the trachea. So we are really studying how whether these observations of mechanical signals activating tracheal remodeling in regeneration are also playing a role in tumorigenesis of the gut. And we are also moving some of our results into a mammalian system, where we see that piezo, which you can see here labeled with an RNA scope. We have done RNA scope to detect piezo in the context of regeneration or in tumors of the small intestine in mice. And so here you can see DAPI is in blue. PCAM1 labels endothelial cells, and piezo1, the mechanosensory channel in mammalians. And you can see that the piezo activation, piezo mRNA signal is very much increased in a context of damage or tumors in the mice, not all in the epithelium, but also in endothelial cells. So we are now just having the first few mice where we are knocking down piezo1 from the endothelial cells in the vasculature and asking what the outcome of this is to the regenerative response of the mouse intestine or tumorigenesis. So now I'm gonna spend the next I still have a few more minutes. I would like to spend the last few minutes of my talk give you a little bit more of a systemic view of gut biology and the work we are doing there, and also some touching a bit on more gut to brain interactions. And so and one of the questions that we are want to address in this aspect is to understand how, what is the mechanism behind the phenomenology of cancer or gut disease associated fatigue and sleep disorders. This is a very prominent manifestation of the gut biology and gut disease. But, there is a lot of reports of, cancer patients or people with inflammatory diseases of the intestine or acute infections of the intestine that have behavioral manifestations, including fatigue, difficulty falling asleep, or poor sleep efficiency, early awakening. So, and these are, in many cases, this is just reported as a phenomenology that in many cases manifests itself before the diagnosis of gut disease. So it's not simply can be seen as a consequence, a psychological effect of knowing that you have gut disease. However, very little is known about what are the mechanisms, the biological mechanisms behind these observations. So we were interested in following this up and really start to understand more of this in the flies, knowing what we know and with the models that we have of various gut diseases. And this is work that has been driven by Sofia Polkoniuk in our lab. It's a postdoc in our lab. So what we have done when we started this project was to ask a simple question. And first is, can we recapitulate the phenomenologies that had been seen in patients with gut disease in terms of the impact of gut disease in behaviors? And so we use different models of gut hyperproliferation, intestinal stem cell hyperproliferation driven by different stimuli in the gut, colorectal cancer-like insults, for example, overexpressions of the oncogene RAS or SRC in stem cells, which we know would drive hyperproliferation, oncogenic proliferation of the stem cells in the gut. We have also inflammatory models of gut disease driven by the chronic activation of JAK-STAT signaling by driving, this JAK-STAT ligand or impaired or the interleukin in the stem cells. But we also have models of chemical damage, for example, feeding bleomycin, which is a DNA damaging agent that also causes the gut to proliferate acutely as part of a regenerative response to this damage. So these are various contexts of hyperplasia in the gut, in some cases more related to cancer, in some others related to physiology like regeneration, or in some other cases related to inflammation. But in any case, in all of these, the commonalities that you have a hyperplastic response, a very strong proliferative response of the stem cells. So we do that by, driving the transgenes in specifically in adults. And to achieve this, we keep flies at a non-permissive temperature of 16 degrees. These are flies with the desired genotypes. So with the crosses and the offspring are kept at 18 degrees. And then we move the flies to non-permissive temperature, to permissive temperature of often 29 degrees to activate the transgenes during the adult stage only and for the time frame that we want, for as long as we want. So we keep a control of when we activate hyperplasia by genetic means. And then we measured sleep using activity monitors that looks like this, where you have individual flies in each of these tubes. Then these tubes will be exposed to a red infrared beam that is going to record the level of activity of the flies, essentially how they move within the tube. And if flies, and this is recorded like it looks like this, you will see some flies move, some don't move so much. And basically, a fly that doesn't cross the red beam for a period of 5 minutes is considered to be sleeping. Yeah. And we have this system in a 12-hour light day cycle. And then the activity is recorded, and certain programs are used to read this activity and translate it into reports of amount of sleep, either during the day or during the night. And the graphs look something like this. And I'm going to show you some more in the next few slides. So basically, this shows the portion of the time that the fly spends sleeping, either during the 12 hours of the day period or 12 hours of the night period. So we start with the different hyperplastic models driven by genetic means. The oncogene RAS, if we overexpress it in stem cells, we drive very significant proliferation of the stem cells. And what we observe is that these flies, actually, if you look at the light blue line, flies that have a hyperproliferative gut driven by RAS oncogene do sleep more. If we drive the sort of inflammatory proliferation in the gut, it also leads to more sleep. Another type of oncogene SARC drives the same type of phenotype. So in all of these cases, hyperplasia of the gut driven by different transgenes, the commonality is that you have a very massive hyperproliferation of the stem cells. In all cases, these animals are sleeping more. So gut hyperplasia drives generally an increase in the sleep in these animals. And this is summarized here. I'm going to give you a little bit of more information about one of the models, because this is what we've been, doing more at the minute, but just to make case that this is a general phenomenon of hyperproliferative gut conditions. So what we basically do is we induce for 2 days the oncogene at 29 degrees. We check that the guts are hyperproliferating. So we take these flies, not the same ones that here, but basically at this time point, after 2 days of transgene expression, we put the flies in the sleep monitor for another 5 days. And we check after the 5 days that these flies still have hyperproliferation, because this is something that gives us a good idea of how the proliferation in the gut correlates to sleep. And then we measure sleep over all this period of 4 to 5 days. This is every day we measure in a different condition, in different genotypes, 2 control genotypes, the RAS overexpressing, the light blue line. And this is an average of the sleep fraction during the day and during the night for all these flies. And consistently we see, most prominently during the day, which is the active phase of these animals, the animals are sleeping huge amounts of time. And this is quantified here in a different way. Total sleep. Sleep during the day, very highly increased, significantly, but not as highly increased during the night. But again, flies normally would sleep more during the night. So consistently, this tells us that the flies that have hyperproliferative guts sleep more. What about chemical damage? What about guts that are proliferating due to external insult, not necessarily oncogenes or inflammatory cytokines, but other type of hyperproliferative conditions, which mimic more sort of regenerative proliferation. And what we can see here is that also that driving proliferation in the gut and by the use of chemicals can also lead to increased sleep. So we have different conditions, different models that we can now explore more mechanistically, and how this sleep phenotype can be explained from a mechanistic point of view. But okay, what does this phenomenon, this is more of the same type of quantification. So what does this phenomenology mean? And so to start more really understanding this and really making sure that this is just not a weird observation of a sleep or a behavioral experiment, we decided to then look at the brains of these animals, because we have a sense, a knowledge of how sleep is regulated, or what are the centers that regulate the neurons that regulate sleep. There are circadian neurons that regulate sleep. There are sleep control centers. They talk to each other through the production of different neuropeptides and secreted factors. And so there are some readouts of circadian activity and sleep that we can actually directly look in the brain, and that will tell us whether this phenomenology that we observe in the behavior translates into molecular changes and cellular changes in the brain. So one of these very important regulators of sleep and circadian behaviors in Drosophila is controlled, PDF. It's a very important peptide that is produced in a subtype of neurons called LNVs, lateral neurons, small lateral neurons. That these neurons are very interesting because they are very plastic, and they actually have projections that are quite open and very arborized and active during the day, but they are more closed during the night. And consistent with that, during the day, these neurons secrete high levels of this neuropeptide PDF, and it is a wake-promoting peptide. And this is what we can see normally during the day in the brain of wild-type flies in terms of the levels of PDF in these neurons. So if we look at the levels of PDF in the neurons of animals that have overexpression of RAS in the gut, so these are guts that are hyperproliferating, the PDF levels in the brain of these animals is significantly reduced. And this is looking at the cellular projections at this level, and is quantified here, this reduction of PDF. And we also, if we look at the cell bodies, at the bodies of these cells, we also see a reduction in the production of PDF. And we also know that it is most likely a production of this hormone, because we have now look at the RNA levels of this, and this is also reduced. So there is an impairment in the normal production of the PDF during the day, when the gut, in conditions that lead to hyperproliferation of the gut. And this is also quantified here. We also look at period, which is a master regulator of circadian activity in brain. And we observe that also the levels of period in the brains of animals that have RAS-driven hyperproliferative guts are reduced compared to control guts. And we also look, for example, another readout of, of an neuropeptide like dopamine that is also associated with waking functions in the control or in the RAS overexpressing guts, in the brains of RAS overexpressing guts. And then we see that the levels of dopamine are significantly suppressed also in the brains in conditions of gut hyperplasia. So, and this is also seen here with heat maps. So there is really a very prominent, not only behavior, but there is also actual changes in the brain that are consistent with the behavioral changes that we see when the gut is hyperproliferating. And we have also used sodium, calcium sensors, the same GCAM sensor that we use I show you in the trachea, to look at the activity of these different, these neurons, these PDF producing neurons that are highlighted here with, dotted lines. So you can see this is in the brains of animals that have been fed normal food, or animals that have been fed with bleomycin and they do have hyperproliferative guts, and the activity levels of these PDF neurons, again, this is during the day, is significantly reduced when the gut is proliferating. So there are very significant readouts that are associated with the waking up that are suppressed in these animals. So altogether, what we know is that hyperplasia in the gut, be driven by tumors or in the context of inflammation or in acute proliferations of the intestine in response to damage, do lead to an increase in sleep or fatigue-like phenotypes. We have done a lot of different measurements in terms of behaviors that help, lead us to conclude this. So we are now doing is we are looking into what could be the gut factors that are influencing this sleep. So we are screening for different factors that we see are commonly change in regenerating guts, and seeing whether which one of them would be playing a role modulating this behavior. What are the signal receiving cells in the brain? Are they neurons? Are they the glia? So we've generated dual driver systems to manipulate independently the gut and the brain, and look at how behaviors are affected. So we are going to look into more, unbiased approaches to look at changes that are happening in the brain, in particular circadian neurons, as a consequence of gut regeneration or gut hyperproliferation. And ultimately, what is the physiological significance of this? Is there like a purpose of increased sleep while the gut is proliferating? And this is something that we want, very interested also in addressing. And we are collaborating with Phil Karpovitz in Canada to translate this type of experiments into mouse models of gut regulated sleep. And I would like to finish by thanking everybody in my lab, our collaborators, for the projects that I've talked about, and our funders. And thank you for your time.
Thank you, Julia. It was an amazing talk. We have now time for some questions. There are already a few in the chat box. I will start. So we have a question by Sylvie Henriques. She says, very nice work, Julia. I understood that in your current model, mechanical damage would activate trachea through piezo. But what do you think is the molecular mechanism activation trachea in response to, for example, pathogenic bacteria? Maybe I missed this part. Thank you in advance.
Thank you, Sylvia, for your question. So it's a bit complex because in the context of pathogenic bacteria, we have chemical signals, which is ROS, and we have mechanical signals, we believe, mainly driven by the death of the enterocytes. That is a consequence of the bacterial, the pathogenic infection. So this is exactly what we are trying to define at the moment. We know that there are some common mechanisms that drive the response to chemicals or the mechanical cues downstream of pathogenic bacteria. So piezo is one of the molecular signals, but then we also have FGF receptor signaling, which is a very important driver of tracheal remodeling in response to pathogenic bacteria. So we believe they are independent, like the signaling downstream of FGF receptor from the one downstream of piezo, but we need to still address this. And the fact that we have some conditions where we do see trachea remodeling, but we don't have ROS will help us to try to, dissect this question further. But yeah, this is very important.
He has another question, one more question if possible, Julia. In your sleep assay, can you distinguish sleep and immobility, and could the latter be, or latter one be the phenotype modulated by these manipulations? Thank you again.
So thank you, Silvia. This is also, it's a very important question. So the activity monitors are they measure activity and sleep, really they are linked. But what we know is that these flies don't have an impairment to sleep, to move while they are awake. Yeah, okay. This we know for sure because we have directly tested their capacity to climb up a tube. Where if you shake them when they are asleep, they can wake up. If we disrupt their sleep, they wake up, and they do, they do sort of follow a pattern of awakening between the night and the day. They just take a bit longer, but they are not immobile flies. This is for sure.
Okay. We have a question by Andy Day. They say, have you looked at adenosine levels in these flies at all? Could the hyperproliferation of the stem cells be driving increased systemic adenosine concentration through increased ATP utilization, possibly increasing desire to sleep?
Thank you, Andy. This is quite interesting, actually. So we have one of the projects that we have in the lab related to the tracheal gut and the oxygen, the fact that the gut and the stem cells need oxygen to proliferate, which is more focused on looking at the metabolic adaptations of the stem cells. We haven't looked at adenosine in the context of sleep, no, but we know that these stem cells are using ATP, are using a lot of ATP or producing as well. They have active OXPHOX metabolism and so on. So it is a possibility that we have not explored yet, but it's interesting, yeah.
Then we have Jenna Dow saying thank you, and Anzela Niraula saying that was awesome, thank you, and I make them my words. I also have a question, kind of linking the two projects. I was wondering if changes in piezo or like promoting the tracheal remodulation would be enough to change sleep.
So we have never done this experiment. So what we know is that the tracheal remodeling is basically upstream of gut proliferation. So you can induce, if we induce damage, but we block the proliferation of the stem cells, we do see that the remodeling is still happening. So remodeling is upstream of proliferation. What drives the gut tracheal remodeling is more the stress signals or signals that are sensors of the damage rather than the proliferation of the stem cells themselves. But we don't know in the context of sleep. Our hypothesis is that in the context of sleep, also what drives sleep is signals that are secreted from the gut in response to the oncogenic stress or the damage-induced stress and so on. But we don't know for a fact because we have still not done an experiment where we induce the manipulations but block the stem cells from proliferating and seeing what happens to damage to sleep. So it's a very good question, Rita. We don't know yet. Part of the
Okay, so I don't think there are any more questions. I think our session is finished for today. I think if people want to stay for a Q&A, it's already 5:10. I mean, if people want to stay a little bit more, they can hang around. I will stop the recording now.